Ja, herzlichen Dank für die Einladung. Well, we will switch gears now, more or less completely, because I'm going to talk about a specific kind of system or idea or concept, and will try to convince you that this concept plays an important role in many areas, and then I will focus on a few applications in the wider range of macromolecular systems, and then try to explain what is going on. So here you have two typical macromolecular systems, and we will come back to these later on. And there's a little bit of work which I'm going to report on, which was done in Mainz as well, and these are the people who are involved in these specific cases, and I will mention them on the way. So the question is, random walks, what is it, and why are random walks of interest to us at all? So this term, I think, was coined in 1917 in a question to nature whether one could solve a certain problem. And this is the kind of starting problem. This is the walk of the drunken sailor. You can see this cartoon in many places. So you have somebody with a little bit too much of alcohol, and this guy is trying to find his way. And the first idea is maybe his steps are really randomly oriented, and so he has to go, he really randomly cruises around. And this is an old picture and an old cartoon, and you might ask, is that still of interest? Well, if you look up Physics Today, September 2019, there were two papers or two articles, one about diffusion, a motion of molecules on surfaces of membranes in the general context of biophysics, and you see again these pathways. And of course, what you then will find out, if you would look at this paper, for instance, these are two-dimensional random walks. Or that is the cover page of that very issue as well. Random walks are also used in the context of calculating complicated integrals, and also used in context of machine learning as well. So this very old and very simple concept or idea actually nowadays applies to very modern scientific questions. So let's start with a very simple example and how this looks like. So a random walk, and we do it now on a square lattice to make it even more simple, consists of steps, B1, B2, and so on that lattice. And each step then follows in a random direction from here, so it has a choice to go here, back, there, and it walks around like this. And this is a very simple example of a 2D random walk on a square lattice. And for that, it's trivial to calculate a number of properties. For instance, the end-to-end vector, this blue thing here, is simply the sum of these little steps. And that tells you already, since they are uncorrelated, that the average end-to-end vector is the zero vector. So the average is right at zero because the average over all these bonds is zero, all the four directions. And the mean squared end-to-end distance here, then it's the sum here squared, is because they are decorrelated, they are independent of each other. It's just B1 squared, B2, and so on and so on. It's just B squared, the bond length, times the number of steps. And this is independent of dimension, that holds everywhere. And this is really a trivial thing, no, so to speak, but it has interesting consequences. So if you, for instance, look at the development, for instance, of the x component of this end-to-end vector, and plot this simply as a function of a number of steps, and these are, I think, 6 or 7 walks where it is plotted for, then you find out that the distribution of the position of these ends is a Gaussian. So it's a Gaussian diffusion process if you look at this in more detail. And this can be applied to many, many different areas. So, for instance, Brownian motion is essentially what a random walk is doing. It's also applied to stock fluctuations. There are many theories related to that. You can introduce a direction, a memory, and so on and so on. And then you can look at stock fluctuations. It's used in the context of chemical reactions. And of course, it means essentially it's one of the major tools in statistical physics. And when you look at this now, this is a random walk out of that very Physics Today of September of a few thousand steps, I think 3,000 if I'm not mistaken. as it looks like here, and this is a 2 million random walk in 2 dimensions. And you can see that essentially this little area here, if you blow it up, looks almost like this, and you can apply all the concepts of self-similarity and scaling and so on, which played an important role in the development of physics, of statistical physics and critical phenomena over the last 30 or 40 years. They show up there again, and if you look up Mandelbrot's book on fractals, you find pictures like this as well. So that is all closely connected with each other. However, if I want to apply this, let's say, to molecular structures, there's a complication which might come in. And if I look now at these 2 examples of a random walk, so this one here and this one here, I can introduce what is called a self-avoiding random walk, namely a random walk which doesn't allow to visit places twice. And again, I can run through the calculations and approximations, but this thing already you cannot solve anymore exactly. So, and you can solve it exactly, of course, in 1 dimension. That's trivial. But in 2 and 3 dimensions, you cannot solve it exactly. And in 4 dimensions, again, you can solve it. Because there, this doesn't play a role anymore, and you recover standard random walk properties up to logarithmic corrections. So you have this here is forbidden, while these 2 are allowed on both sides. And if I now calculate something like the end-to-end vector, for instance, and the behavior of the end-to-end vector as a function of number of steps, here I have that this end-to-end vector squared is proportional to n, the number of steps, while here it's proportional to n, that's the way it's written, n to the 2 nu, and nu is a so-called is a critical exponent, and this nu in 3 dimensions is 3/5, the best value is 0.588 and so on. And in 2 dimensions it's 3/4, and that's exact. So that means that r squared in 3 dimensions is proportional to n to the 1.2, and in 2 dimensions to n to the 1.5. And this has dramatic consequences. So 1.0 and 1.2 doesn't look that much, but has dramatic consequences when you look at the extension of such a walk. So this is a random walk of 1,000 steps. This is the Gaussian random walk, and this is the self-avoiding random walk made by the same computer, same random numbers, and so on and so on. So you see that this tiny, quote-unquote, tiny change of its influence of this exponent leads to dramatically different structures. And this, of course, already looks a little bit like a macromolecule, or one could think of a macromolecule. And I have 2 examples here. So this is a piece of a polycarbonate. That's the material compact discs are made of. Here, these glasses are made of, lenses and cameras are made of, and so on. And this is an IDP, an intrinsically disordered protein. There's a huge class of proteins in our body which do not fold, which stay disordered, and their function is intimately linked to strong fluctuations of their shape. And yeah, at a first glance, they look a little bit similar. And the question is, why are simple random walks or self-avoiding walks good models for polymers? So why do details here not dominate everything, or details here, so that these 2 systems are dramatically different? Or is there a scale or a region where they essentially are similar, or where they are the same? So in the realm of the talk from yesterday evening, these are the kind of systems which are in the middle. No? There are the gigantic systems, there are microscopic systems, and we are living in the middle where all complications come in, and we have to sort out what do we need and what do we not need. Well, in order to understand why this is really a very useful approach, we have to go back some almost 50 years. We have to go back to de Gennes. And so he looked at a standard magnetic model. So you see here an Ising model, no? Spin up, spin down in 2 dimensions. This is the Hamiltonian. You have the spins, the exchange coupling, and the magnetic field. So very simple. And when you can calculate the correlation function between positions here, which then lead to the susceptibility of the system, by doing what is called a high temperature expansion, and that means the correlation between these 2 positions is given by the sum over all steps, which of all random walks, which go from here to there, and with a coupling to the power n, where n is the number of steps. When you write down a high temperature expansion, that's exactly what you get. So you sum over all paths which connect these 2 spins here and there. So this is a simple ansatz in statistical physics. And when you look at the number of components, easing is 1, so that is this one, up and down, the XY model has 2, and so on and so on. And now Dejean had really an ingenious idea. He did the following. He said, he made an analytic continuation in a way that he said, I look at the system where the number of spin components, which is 1 in the easing model, 2 in the XY model, so that means where the spin can rotate on a circle, goes to 0. This is just a formal trick. And when I do this formal trick, then I find that all paths between this spin and this spin, which have such loops, which means where this path visits a position twice, get a weight, a statistical weight of 0. So if you have a statistical model where you formally go to 0 components, you find that only paths survive which are self-avoiding. And this has dramatic consequences, because this is in the context of critical phenomena, allows you to decide about relevant variables. You have critical points, you have scaling laws, and so on and so on. And you can go on, and then you can show that in the case of these walks, the inverse chain length, so the inverse number of steps, is the same essentially as the normalized distance in a magnetic model from a critical point, the point where the fluctuations diverge. And what does it tell us? It tells us that all polymer-like objects, for certain properties, behave the same, completely independent of chemistry. You can forget about the chemistry. The whole chemistry is sitting in a prefactor. And so my next door neighbor in the institute is a synthetic chemist. He loves it if I say this, that I can pack everything in the prefactor he is doing, or she is doing now. But this is, for a description of the systems, this is a very important step, because it allows us to use very simple model to make statements about rather complex physical systems. And that means, here's again our polycarbonate. I can look at the systems either like this or on this very simple level. And what I'm going to do today is I will look at systems on this simple level and then make the connection also to specific chemistries or biological systems. So that means when we look here, for instance, at many different polymers, so the simple polyethylene, that is this infamous plastic bag, polylactic system, which is biodegradable, cellulose, DNA, or protein, essentially they are all chain molecules, and certain properties, like the typical extension of a chain, some dynamic properties and so on, are independent of this chemistry. Because I have this, and the deep theoretical reason is really this mapping onto critical phenomena, which showed that there's a basic physical reasoning for this universality. It's not just our feeling that we make a match of length scales as physicists usually do. It's much deeper and there's much more to it. And of course, if I want to really then go and describe and understand a specific system where the prefactor plays a role, like the local stiffness or friction in between chains when it comes to dynamics and so on, then I have to really link all these scales. But today I will restrict myself to this situation. And this allows us now, we can do it of course anyhow, but this gives us the reasoning also why it makes sense to tackle a really deep mathematical, scientific, and technical problem by very simple models. And the problem I want to look at is this one. This is a trivial thing for everybody of you, but mathematically it's extremely difficult. Now, if you do statistical physics, where you sample all different conformations, let's say, of such a loop, to distinguish these two cases from the very beginning, especially when this becomes more complicated, is not at all trivial. That is a very complicated and difficult thing to do. And of course, in the context of art, this has played, these kind of knotted structures have played a huge role. You see this over and over again, or here in Christian art, you see this Borromean rings, which are a kind of sign for the Holy Trinity. In Ireland, they make this out of that. The Holy Trinity is purity, body, and flavor for beer. But still, this is of course, an interesting motif. And you can go on, and here's a list of some very simple knots. That's the so-called unknot. That's the simplest trefoil, and then you can go on. And this is still a very active field in mathematics because it's very difficult to really analyze and classify complicated knots. And this is the simplest case because this is the case where I have one loop. And if I think of any physical or biological systems, I might have many loops. Some cases I have one, but often I might have many, and I have to try to connect that and understand that or make a rational out of that. And here's an example where this plays a role in very recent biophysics. When you look at proteins, at folded proteins, not the IDPs, the intrinsically disordered ones, you find that about 7% of all proteins in the folded state are knotted. And there are two kinds of knots, so to speak. You can have knots at the surface, and you can find them, and people do not know whether they are important or not. But you can also have knots, so to speak, here in the middle. So you have a folded structure here, a folded structure here, and then here that's linked by a knot. And where people are now rather convinced that this stabilizes the folded structure and certainly steers the fluctuations of these folded units with respect to each other. So looking at knotted structures in the context of polymers, where this is then somehow a knotted random walk, seems to play an important role. And this is a topic in biology which has been discussed in the recent years more and more. But let's come back to a very simple case of a single chain. So if I have an object like this, of course, this is something different like this, and you might say that has nothing to do with a chain. However, if I think of a macromolecular system, I have this situation here, and then I make these pieces here very, very long on all time and length scales which are of physical interest in this context. This piece here not necessarily knows whether these ends are open or not. So it really behaves more or less like a mathematically knotted structure, even though it's mathematically not knotted. It's, of course, it's open. And here's an example of how this looks like. For instance, in experiments, that's a piece of DNA, which is, when you carefully look at it, is a real knot. It's a topological knot, and you cannot simply pull this open. So to do that, you really have to cut it. And there are many simulations on that. For instance, this is from Gargov-Kantor and Meran-Kadar, where they look at such rings and see how they fluctuate, and they did this for a whole range of different objects, complicated knots. But what you always see is, and I will come back to that, you see that the fluctuations of this object seems to kind of pull the knots tight. No? And this is an interesting, it has interesting consequences. And then, as I said already, in the general area of biophysics, these things are discussed in many details, and this is a paper which came out a few years ago, which calls all these pictures biologically motivated knots. I must say I do not know a protein which has a knot like this, but it's a nice picture. And these structures are becoming very complicated and also mathematically extremely demanding. So let's now look at this case again. So when I have, let's say, an object like this, so this is in principle is a random walk, or it's a self-avoiding random walk. I locally have a knot, and then I can ask myself what happens to this knot. This knot here is, of course, a region of small local conformational entropy. So entropy means I count the number of states which the bonds can have. And when you look at this now, the penalty of entropy here has to be balanced with the influence of on the entropy if this knot is not as localized as here, but is much bigger. And what you find then is that if you fix, for instance, the chain ends and let the system fluctuate, that these fluctuations really pull the knots tight. And then depending, of course, on the length, the knots become tighter and tighter, and you can study this in more detail. This has immediate direct consequences. So what does this have consequences for? Well, it has consequences for the way you read DNA. So there is this DNA translocation through a micro or nanopore, which is used, or people are working on it, but I think it's already on the market, used to read out DNA. So what you do is you have a pore, and when you have an electric field, DNA is a charged a polymer in water, it's negatively charged. And you have this pore, and you apply a field, and when counter ions go through that pore in this direction, and the charged chain goes through that pore in that direction. And when you do that, you can directly see whether the DNA has a knot. So this is again the experimental setup. And here you can estimate the percentage of knotted chains and compare this to experiment. And here's how a typical experiment looks like. So you start out here, that is the flux of ions through the pore. When the chain enters, the flux is reduced because this has a finite volume, so less counter ions can go back. You have a reduced flux, and then the chain goes through, and that's it. By this, you can measure the length of a chain, and by certain fluctuations, you can even read out the DNA by now. And here you see the folded chain end coming in, so it occupies more volume, so the flux of counter ions goes down, and the same happens here when you have a knot. So the chain comes in, and when the knot is in the pore, the flux goes dramatically down and goes up again, and when the pore is again open. And by this you can really directly see what kind of knot or how you have, and that the chain is not fully stretched anymore. So this is something which is done now, I think technically almost routine in many areas of biophysics. So that means the knottedness of the structure and the fact that this cannot cut through each other really plays an important role in the way these experimental devices work. Now let's go to a different situation, where we have a huge system, a melt of many chains, and in this melt of many chains, as you see here, that's a simulation of 1,000 chains of length 2,000. These chains behave like random walks. What does that mean? Well, if these chains, this red chain is a random walk, the end-to-end distance squared and the average is proportional to the chain length. You have this Gaussian distribution of the end-to-end vector, just as in this one-dimensional example I showed in the very beginning. It's exactly the same in this context. And what you have is you have a so-called self-density. So when you take the volume of that object, the self-density goes like n, the chain length, to the minus 1/2, which means the self-density becomes very small when the chains become longer and longer. And this has dramatic consequences for the behavior. So when you look, for instance, at the viscoelastic response, so you take such a system, you shear it or you pull at it, you have a very fast decay of your elastic constant, and then it stays constant for a long time and eventually decays. And when you take a network where the only difference is that you have a few glue points here between the chains, you get exactly the same curve with the only difference that it doesn't decay. And the question is, how can one understand this? And that goes back to Edwards already in now more than 50 years ago. He realized if I have something like this, the fact that this is important leads to the fact that these chains only can fluctuate around in a tube-like region along the backbone, because any leakage, like this red one here, would cause an enormous entropy penalty, because essentially your chain is double-folded here and has much less options to fluctuate around. And because of that, the chains should stay in a tube-like regime, and then it was Degennes who realized if I make this very long and this very long, this whole picture holds for open chains as well. And that defines characteristic lengths and so on, which I don't want to go into. The important point is, from this argument, this so-called entanglement length, so the typical number of beats on a diameter of this tube-like regime, this NE, should be determined from conformational statistics, because that is the only thing which goes in. But experimentally, you only have dynamic measurements, indirect measurements to get to that quantity. So you have to connect that to statistical mechanics. So these are mean square displacements I think we do not need. And that this really happens, you can see here, these chains really stay within this tube-like regime. So this is a kind of picture here, and you see that this middle part, for instance, survives all the time, and it resolves these properties from the chain ends. So this is a random walk in the presence of all the other random walks of the chains, with the only constraints that the chains cannot cut through each other. And then you have this kind of behavior. And you can see this also for DNA, where you can directly see this with confocal microscopy. When you have entangled DNA at time 0 and time 1 minute, you see again this opening of a tube from the chain ends and a little bit of tube leakage here, but this is very small. So it's, again, for the standard polymer like a polystyrene or whatever you have, and DNA behave in the very same way, following this behavior of the self-avoiding walks in a melt, which results in these random walks in the melt. So we should come back to get this data or this information out of statistical mechanics. And as I said, the chain has a very small self-density with all the other chains around, and the relaxation time within this reptation model is extremely long. So tau d, with this so-called disentanglement time, is proportional to the chain length to the power of 3.4. What does this mean? That means when you look at a typical tire rubber, which is used for tire production, you can, without vulcanization, you can leave it for a year and it doesn't do anything because it's so viscous. The relaxation time of the chains is so long that nothing happens. It's like a solid, but it's a liquid. And you can try to estimate these entanglements between the chains in many different ways which produce this tube, but that doesn't really help. And the problem is why it doesn't really help is that you not only have a binary interaction between two chains, let's say, you have to take into account the system of many random walks nested into each other and many chain or many walk effects. And you can see this here, especially for the Borromean rings, which I mentioned before. That is this cartoon of this picture of a Borromean ring, and here it's again. And if you take, for instance, the blue one away, then the red and the black one are not linked to each other anymore. And these kind of effects you have to be able to take into account if you want to understand what is going on. Otherwise, you are lost. And there's no way I know of so far to do this really in a clean way mathematically. So for these 3 rings that might work, but if you have very long chains where hundreds of chains share the volume of a given chain, this is essentially or not only essentially, this is at least so far impossible. And this, of course, also holds for open chains. So when you carefully look with these 3 objects, and you make these here very long, let's say, again, along the philosophy of de Gennes, you see that this part here is nothing but this picture of a Borromean rings. And you might have much more complicated situations again and again. But Edwards already had the idea. Edwards' idea was, well, in principle, the only thing I need is, and this is a drawing of Edwards from '67, it's really old. Yeah, the only thing you need to do is you have to fix the chain ends and then pull the chain taut, because then you get, so to speak, the backbone of this tube, and then you do not need all these other complications. And to do this analytically is essentially impossible, but what you can do is, you can do this on the computer easily, and then you can do it as it is required, of course, when you look at the systems, you can do it for all chains at the same time. So that is how this looks like. So you take your chains in the melt, fix the ends, and then you simply pull them straight, taking into account all the others in the surroundings, so that you get these kind of structures. And that is then how this looks like. So this is the melt, and this is just these chains which are linked to the yellow one, and you can see how this pulls that straight, and this walk that is now a random walk with different bond lengths, so to speak, which is close to what is called also a Levy walk in a completely different context. That is the backbone of this tube-like regime, and that you can use to estimate this elastic constant in a scaling type of way. So the elastic constant is nothing but an energy density, this plateau modulus which I showed before, and we measure then this plateau modulus or describe it in kT, where it's the energy scale, divided by the characteristic length scale of a chain, which is a measure for the intrinsic stiffness, times a scaling function. And when you do this, it quantitatively fits the experiment, but what is interesting here is you can use this scaling to plot this plateau modulus for many, many different systems, from solutions of very long chains to dense melts, and for polycarbonate, which I mentioned before, there's this point up here. And there's not a single system we looked at which really was significantly off that curve. So in principle, you can, by using these concepts, you can directly infer the entanglement length and the properties just from a few input parameters which one knows. And one can then go on. This is the region of entropic elasticity, and when you go on to energetic elasticity, so these are microtubuli in cells, for instance. You have a unique curve now over 7 decades here, 8 decades here, and 5 decades here. And these snakes are somewhere here. If you take the characteristic length scales of the snakes, you can put them on the curves as well. So there we have a reasonable understanding by employing all these concepts, and we can, of course, ask now what happens if I take that very system and pull at it. Huh? And I can do this again and see what happens, and that is how this looks like. And then I can ask myself what happens. Well, I start out with a primitive path like this, and the unperturbed melt. When I pull at the system fast enough that the chains cannot relax, which is experimentally the standard case, and I can ask myself what happens here, and what I then see is that I find something like this. And this is something which we had before. No? You remember in the very beginning I was talking about pulling a knot tight because of entropic fluctuations. That shows up here again, and in principle one can again apply this picture. It's not absolutely clear because it might also be a Frenkel-Kontorova scheme, and this is one of the interesting questions. It really delicately depends on local interactions. So Frenkel-Kontorova is essentially, the original paper is you have a sinusoidal well where particles are in, and when you pull at the particles, and then you, in the end, arrive at a system where you have dense and very dilute regions because of a nonlinear elasticity of these chains. But this is an interesting side remark. Now, what I was always talking about this situation. But you can also have this situation. And of course, you can ask whether a theoretician's playground, so you make a melt like this and look at this. What the hell do we need that for? Well, we need it for certain aspects. For instance, if you take a hydrogel, which is used in many hygiene products, for instance, but also in food and in biology in general, if you shrink such a hydrogel, so these are the chains and there's water around, and you shrink that, you see that these chains do not really overlap, interpenetrate like in a melt. They stay segregated. And there's a fantastic example of segregated chains. That's this one. That's this famous picture of the Kremer brothers. So we are not related. Their name is written with C, my name is written with K. Where they discovered what is called chromosome territories. So that's the interface nucleus of a chicken, and you see that these different chromosomes, here are other pictures, occupy territories. So these long chain molecules in the cell nucleus are collapsed random walks on the first site. And this is something which at the beginning is extremely surprising. Why the hell are these chains collapsed? Why are they not internested like in a regular melt, which one would expect? Now you can ask yourself, not necessarily how this is done, but you can ask, why does it make sense? There's a nice paper by Ralph Everers where he estimates that, and that's summarized here. If you take the typical entanglement length of DNA in the interface nucleus, for instance, of a chicken or of us or so, you can estimate the entanglement length. So it's a fairly rough estimate, but nevertheless, you take the size of a nucleus, you take the density, and then you find that even the shortest human chromosome has about 1,000 entanglement lengths. And now you can try to estimate how long would it take for this one chain to relax. For this one chain to relax would be so slow that we could not live. So the estimates go between 50 and 5,000 years, which means a single mitosis would take a few hundred years. That would make everything a little bit too slow for us. No? So there's a good reason that the chromosomes are segregated. Now, is there a very complicated biological apparatus needed to do that or so? That is something physics can ask, and the simplest thing to look at is, for instance, to look at a melt of such rings, so random walk rings, where you do not allow them to concatenate. You can go through the statistics, and you can go through a lot of analysis, and I just want to show you one picture. If you take, let's say, 50% of the chains out, that is a melt of open chains, and that is a melt of rings, you see that these rings there also occupy territories. And they behave very similar to the biological system. So you can look at the self-density. That's what you expect. So the self-density becomes independent of chain length, which means they have a constant density in the territories, unlike random walks and so on. And then you can ask yourself, as a physicist doing such a simulation and doing some estimates, you have a simple system, but is there any relation to what happens in biology? No? You can look at a quantity which is discussed in detail in fish and high sea experiments. And what do? These experiments measure the probability that two pieces of a DNA are close to each other in space, in the cell nucleus, as a function of the contour distance which is in between them. And when you look at the simulation data, you get curves like this, and there are scaling relations for so-called exponent beta and then gamma. I don't want to go into detail here. And what you find is that, for instance, for human chromosomes, here, these curves here are very close to these simulations. So the structures seem to be fairly close to what we have in biology. However, there's one outlier, and that is yeast. Yeast has about as many genes as we have, interestingly. But the chromosomes seem to have a completely different structure. And the point is, when you look at yeast, the non-coding pieces in between are much, much shorter. And when you really do the calculation of chain lengths, you find that the yeast chromosomes are so short that it's not needed to be segregated. Their relaxation time is so fast that you easily can match the biological situation. Okay, there are many interesting questions about the dynamics of these systems, which I want to simply skip. The only thing I want to show you how such a collapsed object moves around, and this chain is twice as long as this one, same magnification, and you see that this really is this very fast fluctuation, but it doesn't really open, it stays segregated because there are all the other rings in the environment as well. You can go on and then make a detailed analysis of threading and to what extent this is important, and that leads to new views also on the glass transition. But let me simply stop here and thank you very much for your attention, because he got already up.